Good morning once again. My name is Leifils Vedaste N, a biology teacher. I welcome you again. I think you remember last period we talked about compound light microscope as a device uses a beam of light to give a magnified image of object. Then we talked about magnification and resolution. Magnification means increase in apparent size of object, while resolution means ability to distinguish two separate points. So today we are going to describe electron microscope. Electron microscope is most powerful device than light microscope. Electron microscope uses the beam of electrons instead of beam of light. Electron microscope uses the beam of electrons instead of beam of light. Question is there any importance of using beam of electrons instead of beam of light? The answer is yes, because electron beams have a much smaller wavelength than light beams. This permits to produce higher effective magnification than light microscope. I repeat, electron beams have a much smaller wavelength than light beams. This means electron microscope can produce higher effective magnification than light microscope. Now, what are the types of electron microscope? One is transmission electron microscope. Transmission electron microscope. Two, scanning electron microscope. Now, let's try to describe each type of electron microscope. One, transmission electron microscope. TEM. Transmission electron microscope is used to study or to describe the internal structure of cell. This means it illustrates urethral structure of the cell, used to describe or to study the internal structure of cell. Electron beams pass through a very thin prepared sample. The specimen must be supported in resin block to prevent collapsing during cutting. I repeat, specimen must be supported in resin block to prevent collapsing during cutting. Then the specimen is sliced with diamond or glass knife. Finally, the section is then impregnated with heavy metal stain such as osmium tetroxide. I repeat, the section is then impregnated with heavy metal stain such as osmium tetroxide. As a beam passing through the specimen, what happens? 1. Electrons are absorbed by heavy stained parts. Second, electrons pass readily through the lightly stained parts. 3. Electromagnets bend the electron beam to focus on image onto a fluorescent screen or photographic film. A photographic taken through an electron microscope is called electron micrograph. I repeat, a photographic taken through an electron microscope is called electron micrograph. The most transmission electron microscope can distinguish objects as smaller as 0.2 nanometer. I repeat, the most transmission electron microscope can distinguish objects as smaller as 0.2 nanometer. This means they can produce clear image magnified up to 250,000 times. I repeat, they can produce clear image magnified up to 250,000 times. Now, the final image produced is two-dimension image. Two-dimension image. Notice, the magnification is varied by changing the strength of electromagnets. I repeat, the magnification is varied by changing the strength of the electromagnets. Second, let's describe scanning electron microscope. Scanning electron microscope, SEM. Scanning electron microscope is used to produce three-dimensional image, 3D. I repeat, is used to produce three-dimensional image of the surface of the specimen. Electrons are reflected from the surface of the specimen stained with a heavy metal. This enables or allows the scanning electron microscope to produce images of whole specimen, for example, cell, tissue, or organ. Notice, electron microscope has not completely replaced light microscope because light microscope used to examine living and unstained specimen. Transmission electron microscope and scanning electron microscope generally they use the beam of electrons because the beam of electrons have short or smaller wavelength so that they can produce the clear image. But transmission electron microscope produces two-dimensional image, while scanning electron microscope produces three-dimensional image. Another point, how to prepare a slide, permanent slides. Mainly, what is a slide? A slide is a small flat piece of glass on which specimens can be mounted for microscopic study. A small flat piece of glass on which specimen can be mounted for microscopic study. General preparation of specimens for electron microscope is complicated, very expensive, time consuming. Preparation deals with dead specimens stained with heavy metals. Now, preparation of the slide involves different techniques such as 1. Fixation. During fixation, tissues must be killed rapidly. Original structure are maintained. Tissue is hardened so that this section can be cut. Chemical used called fixative. Second, dehydration. Removal of water to prevent decaying of material prepared. 3. Cleaning. Alcohol is used to clean the medium prepared. After, alcohol is replaced with cleaning agents such as xylol to make material transparent. 4. Embedding. Very thin sections can be cut if the materials are embedded in a supporting medium. 5. Sectioning. Materials are cut into very thin sections. This allows sufficient light to pass through them. Materials used to cut can be razor or microtome. 6. Staining. laser or microtome. 6. Staining. Use the chemical to change the color of the specimen prepared. Allow the specimen to be seen clear. For example, acetic or cyan stains DNA dark red. Another example, Janssen violet stains bacterial cell wall violet. 7. Mounting. Covering stained sections in a resin medium. This excludes air and protect it permanently. Thank you for hearing and watching this video. See you next by demonstrating questions and answers about microscope in general. See you.